So first, of course, I have to say thank you for inviting us here to talk to you today. It's our second time back, and certainly thrilled to be here and see all of you. So I'm Hilary Savoie. I'm here from GRIN Therapeutics. We're going to share today with you some of the work we're doing in our program developing investigational radiprudil for GRIN-related neurodevelopmental disorder. I'm going to start today with some background about GRIN Therapeutics. I'm going to spend a little bit of time pausing and talking about the advocacy function, because I think this is a really helpful just awareness raising discussion to have with this community, and some discussion about clinical trial readiness and all of the work that we can do collectively to be clinical trial ready. And then I'm going to hand it over to my colleague, Russell Chin, who will be sharing the results of our Honeycomb trial, which is our phase 1b clinical study in children with GRIN-related neurodevelopmental disorder gain of function variants. So who are we? Some of you know us, some of you don't. We're a New York City-based biotechnology company. Our parent company, Neurvati Neurosciences, which you hear often Neurvati GRIN together, oversees GRIN Therapeutics with full funding from a single investor through approval. The company was inspired actually by many of the people in this room, actually, inspired by a 2019 GRIN conference at Emory University. And at that point, in 2021, a partnership with Neurvati and the formation of a patient-focused leadership team happened. We are developing investigational radiprudil for the potential treatment of GRIN-related disorder, as I said, with gain-of-function variants in the Honeycomb trial. Our initial recruitment has been completed in the first trial, and our top-line data was disclosed at a scientific meeting earlier this fall, actually 2 of them, and the study is planned to expand in 2025. We're working very closely with the entire GRIN community. Again, many of you in the room, thank you so much. And we're working really hard to ensure that all of the needs of the community are closely aligned with our trial objectives. I hate this slide because you get to see me twice. It's a little awkward, but this is me. I'm Hilary. So I'm the Director of Community Engagement and Communication at GRIN. And I want to mention this as the head of advocacy to be what was effectively one of our first hires day one of the founding of the company. It's unusual. And I believe that this really speaks to how closely aligned our thoughts about the community as a whole are to everything we do. And my clinical colleague here can attest to the fact that I put my nose in all of the work that happens at the company. Prior to joining GRIN, I founded and ran the Cute Syndrome Foundation, a nonprofit that was focused on another rare neurodevelopmental disorder. I have a PhD in communication and rhetoric. I don't know how I landed in biotech, but that's how it goes. Although I guess maybe I do know how I landed because I'm the mom of a little girl named Esme, who has an undiagnosed rare neurodevelopmental disorder. So she kind of made my life take a left turn. So as I mentioned, I got my start in the rare disease space as the head of an advocacy group and then transitioned to industry. When I joined GRIN, I did so because I really thought that there was a ton that I knew as a parent advocate that could help in drug development. And that is absolutely true. But I also sort of realized that there was a lot about drug development that I didn't understand as a parent advocate. And so I want to just share a little bit about what I've learned, bring you under the hood a little bit on a couple of things. I think the last time I gave this presentation, I said we could have titled it Things That Hilary Should Have Known or Had to Learn the Hard Way. So just to bring you under the hood a bit, how the advocacy function works in drug development in general, it's really important to differentiate between an internal advocate who works in industry and advocates like Sandra, who you've seen up here today, who works independently for the community. My role as an internal advocate is to represent the patient voice internally across functions at our company. And I can really only do this well if I engage the people in this room, the individuals surrounding a disease. And it's also to help ensure that we as a company are leaders in how we bring forward treatments to communities. So again, I do this by representing that patient voice across functions, and by really understanding where all of you get your information, what you like, how you like to be engaged, what your unmet needs are. And all of these efforts work to support our trial, of course, and any future trials. But I think it's also very important to understand that the goal of the advocacy function is to leave the community stronger for having been in it, regardless of whether there's participation in our trials, regardless of the outcome of our trials. So one of the pieces of my job is making sure that the people in this room are prepared for trials and understand how they work. And I think that I just want to take a moment to ground us today in the players in a trial. Who is involved in these teams and how they talk to each other, how they work together. Because these terms are often tossed around when you hear about trials, but no one stops to explain how they interact. So if we start over here with a sponsor, that's what you call someone like, an entity like GRIN Therapeutics that is designing a study and carrying it out. So a sponsor's responsibility is to design the study protocol and the instructions associated with the study. Our responsibility is to train the sites that carry out the study, and we'll get to sites in a minute. It's to work with advocates like Sandra to understand the community needs and their feedback, and it's to communicate with each of the sites. So I use this term site, and this one took me a while. When I was an independent advocate, people talked about sites. I had no idea what they were talking about. A site or a primary investigator study site, sometimes you say PI, these are the people who actually carry out the studies and the locations where those studies happen. So a study site or a primary investigator's responsibility, their job is to select, screen, and consent patients. They are the medical decision makers for the patient, not the sponsor. Their responsibility is to carry out the study as designed in the study protocol and the study instructions. And it's to communicate with both the sponsor and the patients and families in the study. We come down to patient advocacy groups. So again, like GRIN Europe, a patient advocacy group's role in a clinical trial is to educate the patient and family community about the clinical trials that are available to them. It's to understand the patient and family concerns that they have, their needs, their barriers to participation, and then to communicate with the sponsor to help us do a better job for the families. And we do that through things like ad boards that advocates in the space have joined us for, that help provide feedback to us. And then the most important people. Most important people are the patients and the families in the study. And in this case, as in any pediatric indication, it's the parents, caregivers, who are making decisions for their child. And their job is to be informed, to consent to participate, to follow the study and site instructions, to understand the decision to join the trial and what that means, to respect trial privacy, and to communicate any concerns that they have to the site. So, I guess, as I said, to the patient advocacy groups, but really to the site, because this is where they're receiving their care. This is all set up because, like everything else we've seen today, or most of everything else we've seen today, this is an experiment. And there are rigorous rules in place to make sure that the results of the experiment are valid and that we can proceed for the best interest of the community. Okay, and so each of these players, though, they all have a very, very different view on the trial. And that gets to some complexity. They don't always speak the same language. So one of the things that we've been doing is trying to think really creatively about how to engage across stakeholders in order to improve the trial experience across all of our studies. And so we've been having these site visits that we do where we bring advocates to actual sites in the study and with site staff and sponsor representation. And we kind of walk through what happens in the study, what the spaces are, what the materials look like. And we're able to kind of learn from one another and get the advocates in a position where they can better educate the members of the community. They can provide better feedback on what's happening at the sites because they can actually see what's happening. Because if you haven't been in a clinical trial before, you might not know what questions to ask or what to think about or what might be uncomfortable for families or ways to make it better. And these have been really, really wonderful learning moments, I think, for everyone involved. And it's something that we're really committed to doing for this community and the other communities that we work in. So that's a bit of background, and just wanted to give that as context for as we talk about this study and the study that Russell will tell you about in a moment. But I want to just start before Russell comes up and take a moment now to explain why we're evaluating investigational radiprudil and GRIN-related neurodevelopmental disorder. We've heard a lot of talk about gain of function and loss of function today. I'm going to start here. Everyone has their way that they describe it. And I love that we have lots of ways of doing this because everyone's minds work differently. But for me, it's a gate, right? So I always imagine the receptor on the cell membrane like a gate in a fence. And just like you have a gate that works totally fine and you never think about that gate, great. You can keep your chickens in your yard if you have chickens. You can have a gate that gets locked and won't open, or that gets sticky and doesn't open all the way. And that is like a loss of function. You have trouble then moving your chickens from, I guess this is 1 yard out into another barnyard area where the chickens need to be. I don't know why. Or you can have a gate that has a broken latch and it just keeps flying open and your chickens escape. And to me, so this is, having an inhibition of the flow or having more flow than you need. And a lot of the discussions that we have in this community are around gain of function, loss of function, because they are so important when you come to think about precision or potential precision treatments for disorders. And our study, as I said, is focused on gain of function. And that is because, here's another drawing of the receptor. And as we all know, I'm not telling you anything you haven't heard today, GRIN genes encode for the NMDA receptor subunits. And one of those subunits is NR2B, or now more commonly referred to as GluN2B. Radiprudil, our investigational compound, is designed to target the NR2B subunit, and it acts as what's called a negative allosteric modulator. So that means, and we've heard about other negative allosteric modulators and positive allosteric modulators today. This means that it's meant to bind to the receptor to reduce its overactivity. And you can see its binding site is here on the outside, not in the pore. And the hypothesis here that we're working from is that by binding to NR2B, the overactivity may be reduced with the potential not only for fewer seizures, but for improvement in neurodevelopmental symptoms. And I guess with that, I can hand it over to Russell to talk to you a bit about the results from the trial.
Okay. So thank you very much. It's a pleasure to be here and to share with you the top line results from the honeycomb study. So like Hillary, I don't really like this slide either. I think it's, but I can contribute it to Johannes for any Asian controls. I hope a control. But just a little bit about me. I'm a clinician. My background is actually in adult neuromuscular disease. and I joined Neurvati GRIN in April this year. So previously, my work was in peripheral neuropathy, and then I did some work in spinal muscular atrophy. So it's a real pleasure to be part of this team and to meet many of you here who are really on the front lines of this work. It's been really educational and inspirational. So we're very happy to share this. As Hillary said, we presented this at ILAE in September, and then as well as at the Epilepsy Foundation Pipeline Conference also in September. Honeycomb, as you know, is a multi-center open label phase 1b add-on study. So add-on indicating that patients could be on other concomitant ASMs, but they had to be on a stable dose for at least 1 month before screening. They had to have failed at least two ASMs. So we are assessing GRIN-related neurodevelopmental disorder caused by GRIN 1, 2A, 2B, or 2D gain of function variants. We have two groups: a seizure group who needed to have at least one weekly countable motor seizure in at least one a week for 4 weeks. And the other group, the behavioral group, were patients who did not meet that cutoff and had a CGI-S, the clinical global impression at baseline. They had to have a score of 4 or greater. So our primary endpoints were safety, tolerability, and radiprotol plasma concentrations. And our key secondaries were related to seizures, primarily with the seizure reduction, the median reduction, as well as looking at some of the key secondary endpoints also included the seizure-free days, as well as the clinical and caregiver global impressions of change. The sample size, the target sample size was 24. We did have a pre-specified data analysis to be performed when 12 or half of the patients had completed the first part of the study. And so we had this data cut in July. At that time, there were actually 15 patients available, 12 of them having completed the first part of the study. Here's a schematic of the design. We had the screening period. As I mentioned, there was an observation period of 4 weeks. Then the patients in the titration period were titrated based on exposure and assessment by a safety review committee. They had very individualized analysis of their PK levels. We had a target receptor occupancy that we were going for. So overall, the median for the titration period was about 8 weeks. This we had initiated at 0.05 mg per kilogram twice a day, and then titrated it up to the target level for the very individualized for the patient. The maintenance period was 8 weeks, and then they continue in a long-term extension period. Patients were given the solution of radiprotol by mouth or by feeding tube twice a day, as I mentioned. Here's the baseline demographics for these 15 patients. We had 8 in the seizure cohort and 7 in the behavioral cohort. You can see the seizure cohort is a little bit younger, for the mean age 5.1 and 7.1 for the behavioral group. A little bit more females than males. And you can see the genes, the distribution is fairly, it's fairly similar across GRIN 1, 2A, 2B. We did not have any 2D patients. The baseline 28-day countable motor seizure frequency, here you could see for the seizure group, it was 25.5, and very wide range from a minimum of 5 up to 86. The patients were, as I mentioned, for the behavioral group, they had to have a score of 4, at least 4 on the clinical global impression scale. So most patients there were score of 5, 6, or 7, and in the seizure group, they were in the severely ill or most extremely ill categories. These patients have been on, the seizure group have been on multiple ASMs, and the mean number of concomitant ASMs was 3. We did not in this 1B study, we did not set a limit on the number of concomitant ASMs. So as far as safety, it appears to be generally well tolerated. This drug has been tested extensively in adults for neuropathic pain. So we had an extensive experience in adults. We're very limited as far as pediatric population, but we're very pleased to see that it's been well tolerated. There were fever, diarrhea, and respiratory tract infection were the most common treatment emergent adverse events. Three patients experienced SAEs or serious adverse events, but all were unrelated to radiprotidil treatment per the PIs. And this listing here, these are the TAEs that occurred in at least three of the patients. So this is the lead up to this slide. Probably this is the one that's most interesting for everyone, including, and we were very encouraged by this data. So the seizure group there, as I mentioned, there were 8 patients, but actually one of the patients was still in the titration period. So when we look at the 7 who had completed the part A or completed the titration as well as the 8-week maintenance period, we see that the median reduction was 86% when you compared the maintenance period to baseline. And the numbers that actually were over 50%, we were 71% who did that, who had a reduction, 442 greater than 9% reduction, and we had a single patient, or 14.3%, who had 100% reduction. There was one patient here, the gray box, this patient was the one who had a very low number of seizures at baseline, had 5 seizures. So went from 5 to 7. So that's explains this 50% increase, but they had a very low baseline to begin with. We also looked at the seizure-free days. And so in the maintenance period, we see that 6 out of the 7 had at least 80% of the days of the 8-week maintenance period, they were seizure-free. You can see across all this improvement from baseline to the maintenance period. And then getting at the behavioral cohort, we have this early data. This is, again, the clinician and caregiver global impressions of change. And so on the left, the clinicians rating the seizure and behavioral group, and the similar on the right. The takeaway here is that the majority were reporting, both clinicians and caregivers were reporting minimal to much improved status here. So this we found was a positive trend, a positive direction in getting at things beyond seizures. So in conclusion, radiprotidil appears to be well tolerated, and in this patient population of people with GRIN neurodevelopmental disorder with gain of function variants, the most common adverse events were associated with infection or underlying disease. We saw this median of 86% seizure reduction, and most patients actually had 50% or greater reduction, and seizure-free days were seen in 80% of the days in the maintenance period were seizure-free for 6 of 7 patients. These clinical global impressions of change are improvements observed across both groups, regardless if they had qualifying countable motor seizures or not. So this, in sum, this data has been very encouraging, and we are currently in talks with regulatory agencies about the next step to move towards a pivotal phase. So with that, I will just hand it over to Hillary.
Stay right up here, I think. Okay. All right. So Russell talked us through the study, and we've talked about some of the specifics of advocacy and clinical trial readiness. But I just want to underscore again that the broader community is so important in this process, and there are ways to be involved regardless of whether you intend to be a part of a clinical trial or can be a part of a clinical trial. So, thinking about how we get involved. The first thing we're doing right now, right? It's showing up, being here, having the conversations, learning whatever you can learn. Being aware of what's happening in the community for trials. So again, you can read about the honeycomb trial or any other clinical trial that is enrolling in the space at the UDRA-CT website or clinicaltrials.gov. It's a great resource to learn more. You should all be staying connected, of course, with advocacy groups for updates on clinical trials and any other research that's ongoing. Again, we've seen today the importance of that research. And to participate in efforts to better understand GRIN-related neurodevelopmental disorder. Again, we just heard from our colleagues in Leipzig and the work that they're doing and the importance of being sure that your information is in the registry so that people know who you are, where you are, and can learn more about this disorder. And again, you're welcome to check out our website, and you can contact us at patient advocacy. That goes directly to me. We're a pretty small company, so I receive those emails and look forward to hearing from you. I know it may not always be apparent how important it is, your engagement. Often you feel like you're just one person, but it is so crucial that people are engaged, involved, stepping up and getting involved in any way that they can. So with that, I just want to say thank you again to GRIN Europe for inviting us here to speak today. Thank you to all of the scientists, collaborators, investigators who've been involved in our studies. Again, some of you in the room. I can never say thank you enough to people who participate in not just our clinical trial, but any clinical trial. Thank you. And thank you to the GRIN community for providing encouragement and support. Thanks.